the interaction that stabilizes the alpha helix. What are these interactions? So what do you think? Let's start one by one. A, hydrogen bonds between several parallel alpha helices. What do you say? No, this is not correct. Hydrogen bonds between chemical groups in the protein backbone. So this is correct. And you have also hydrogen bonds between protein residual groups, and hydrogen bonds between several perpendicular alpha helices. So what is the final answer here, you think? B, right. Okay, this is about the two dihedral angles that we defined last class, was it? And one before that. And so every dihedral angle in the polypeptide backbone has a certain range of possible values. It's maybe a quite kind of a soft question. I wouldn't say it's the best for an exam because it might has. I'm not sure about a definite single answer, but let's discuss it. No, so the question is, so the comments, not the comments, the guide, there are no, so the guiding instructions are that when you have no question, then just mark the correct or the most correct answer. So there's a relatively large range in the proximity of a large amino acid. Is this sentence is correct or no, or not? So it's incorrect. If at all, it's the opposite, right? B, there is a relatively large range in the proximity of small amino acids, so this is correct. I would say, in principle, hypothetically, and all that, but still, this is the most correct sentence, and the same for the for D. The range is small. Sorry, for the range is large for long. No, sorry, for a short. The range is small for a short polypeptide, and the range is large for a long polypeptide. So the only answer here is B. What about C? The range is unaffected by the nature of proximate amino acid. Well, again, if B is correct, then C is definitely not correct because they are. What do you think? Itself, yes, yes. The range is unaffected by the nature of a proximate, meaning not the same amino acid, but the proximate amino. This is what you gathered from this sentence. So it's not phrased correctly. That's the proximity. I mean, a nearby amino acid. Well, I would say that it might have a small effect on the dihedral, but okay, we didn't discuss it. And no, C in this sense is, as you say, it's a correct sentence as it is. But again, I would discard this question if it will find its way to an exam. It has too many ambiguous points in it. Yeah.
So if
are you sure?
Because
what?
So we are talking only about the amino acid itself and his residual group, and not about the amino acid nearby.
Right.
Okay, so everything about the proximate is, if it's on the amino acid itself, then okay.
So as you said, you're definitely right in the sense that, I mean, in the scope of the discussions that we did, C is correct. That's my answer here. But the question itself is, it's okay. It's good that you try to answer it, just to go over the material. But as it is, it's not a validated question. In proteins, structural motifs exist. So this sentence is just to get you into the environment, to get you into the right mood to answer this question. And now mark the incorrect sentence. So motifs are different combinations of secondary structures. What do you say? This is correct, so you shouldn't mark this sentence. B is a motif implies the protein function. So generally that's true. C, a motif will include the exact same sequence in different proteins. Well, not really, because that is kind of the definition we gave for the consensus term, which is consensus is when you have a high degree of similarity compared to random sequence that you'll choose. between different sequences or different sequences of amino acids, different sequences of nucleic acids, and so on. But a consensus is a high degree of similarity and usually not an identity. And a protein can have more than one motif. This is also a correct sentence, and the incorrect is C. So the correct answer here is C. 4. In homologous proteins, there is. What is the relation between homologous proteins? This is kind of the question. An identical sequence of amino acids. So what do you think? Again, it's the same issue of consensus. So a similar sequence of amino acids compared to an unhomologous protein. So B and D here are correct. So that means that BD, BBBD, H. Okay, 5. Chaperones are involved in proper folding of proteins. Mark the correct sentence for most proteins in the cell. So A, chaperones fold proteins to a thermodynamically unstable shape. This is not correct, incorrect. Chaperones fold proteins into a shape to which the protein is unable to acquire spontaneously. So this is also incorrect. And chaperones they can be addressed as enzymes that promote some chemical reactions. This time, these enzymes promote the correct and rapid folding of proteins. This is the way you should look at it. C, chaperones promote proper folding, which is correct. Chaperones slow down proper folding, which is incorrect, and we stay with C, the only correct answer here. Okay, an enzyme's active site. So this is just a question about definition we gave in class. The active site of an enzyme is its ligand binding site together with the catalytic site. So A and D gives F. Okay, and last question about chromatography. So passing of protein mixture through a column, this is what we call chromatography, is a technique for protein separation. What are the features that can be used to separate proteins in this technique? So what do you think? A is charge, that's correct. B is size, correct also. We gave this specific 2 examples. And placing the cell and involvement in a molecular pathway, well, it's kind of a tricky question again, because sometimes sometimes proteins that are localized in the same compartment in the cell would have a similar domain or a similar motif, and then you can sort of think about an antigen that will be more affinity, that will grab in high affinity these proteins and will isolate them from other proteins. But as a whole, it's a very ineffective. I mean, trying to isolate proteins by their location in the cell using this technique or by any specific domains that it will have is not the main goal or the main the main goal of chromatography. And charge and size are the 2 features that are definitely part of this technique. So A and B, and that means G. Yes.
What is molecular pathway?
Okay, molecular pathway. We really didn't use this term. It's and we will use it in future classes. So first, you can Google it, and I will try to give the first sentence of what Google will give: a bunch of proteins that are involved in the same specific function in the cell. For instance, ATP production. For instance, calcium buffering in the endoplasmic reticulum. I can say many pathways that I know of, and so if they are linked together in a very direct, closely related manner, they can be termed a molecular pathway. And it's a pathway, not only a group, because we sort of know the interactions between them. We know that X influences Y, that influences Z, and so on. So we kind of we see them as a pathway. Okay. Yes.
About question 3.
Yes.
You said that a motif implies the protein function is correct.
Yep.
but we said that in protein we have several motifs, and every motif implies for a certain function.
So that sounds weird to you. A protein can have more than one motif, so then a protein can have more than one specific function. And by function here maybe is the wider, it's a function in the wider sense. So first of all, let's recall what we discussed in class. A motif, even the examples we gave, we attributed to each one of them a specific function. The calcium binding motif can buffer calcium, can store calcium ions, and so on. The RNA binding motif. So a function for the RNA motif, for example, is binding of RNA molecules. It's a function. And so I mean, it's one of the, maybe not the,
When you were talking about the
yeah.
protein function, you're talking generally talking about protein.
Yeah, I see what you mean. Okay, so when we said protein function, I agree that we usually meant, so for instance, the protein function of pKa is to phosphorylate other peptides and not just to bind other phosphates, other polypeptides, because it will definitely have some other polypeptides binding motifs in it. Okay, but I think it still holds. Okay, so just maybe make a distinction between. I mean, we use several words as definitions or terms in biology, and other words as just words. So function here means exactly that motifs, as it's written right here, motifs imply or motifs give some function or some feature or characteristic to a protein. It enables it to grab something, to be somewhere in the cell, and so on. In this sense, it's a function. And if it wasn't an exam, maybe I would give a clarification while doing the exam. Okay, it's okay. Okay, we have a new laser. Very exciting. So we move on to the biomembrane and cell architecture chapter. So it's about the okay, pretty small, the laser. Anyone see the laser?
Yeah.
Not really. Okay, if I were reading it, I'll use the mouse. This chapter is about the characteristics and structure of biomembranes. We've already discussed or presented the biomolecule, the building blocks for biomembranes, the lipid, and we will add more information about that. And it's also a good time point to go over the various cellular organelles. So we'll do that also in this chapter. And this chapter will serve as a background or as a solid ground towards the next chapter, which will be a general discussion on intracellular signaling. So in intracellular signaling, we actually pass ions and other molecules across cell membrane or this barrier. So today's class is about exactly this barrier, the biomembrane, and this is how it links to future classes. So a few terms, even before the overview, as a warm-up. So eukaryotic cells. Eukaryotic cells mean cells that have a nucleus. We are built from eukaryotic cells. The neurons are eukaryotic cells. And they can, if you look at them closely, you'll see some compartments in them with specific structures and specific functions, and they are called organelles, cellular organelles. Each of these organelles is surrounded by a membrane. And has in it a specific set of proteins that allows it to perform its function. An aqueous interior that will be found inside an organelle is called a lumen. So several organelles in the cell have such lumens in them. And the dense network of filaments of structural proteins that exist really anywhere in the cell, everywhere in the cell, and it can be referred to as the skeleton of the cell or the cytoskeleton. So they are, as a whole, they're called the cytoskeleton. So in this class, we'll go over the characteristics and composition of biomembranes, go over the several eukaryotic organelles, the cytoskeleton, and end with a few words about the methods we have to purify and isolate and characterize a little bit some of the organelles and cells. And we will use here some of the methods we've already discussed in the context of protein structure and function. So this is really a recall. You should be able to understand the following sentence, sorry. In the phospholipid bilayer, polar groups face outward to shield the hydrophobic fatty acyl tails from water. So this sentence summarizes the structure of the membrane, which we have already explained. What are the interactions that stabilize the biomembrane? The interactions between the lipids. Let's start with this region, the hydrophobic region. What interaction would you find here? Would you expect to find here? Between the fatty acyl hydrophobic tails of each lipid. The weaker, that's the final guiding question I'm going to give, and then I'm going to give the answer. So the weaker interaction that exists. Okay, so van der Waals is the weaker, so that was my mistake here. The hydrophobic effect, and also van der Waals interaction. So these two interactions can be found to stabilize the biomembrane. But the more intuitive thing I would say is the hydrophobic effect. Between this highly hydrophobic region, you absolutely would find this interaction, and also van der Waals, simply because the molecules are very very close by in space, and this is exactly where van der Waals interactions go into play. And this hydrophobic region is an impermeable barrier to hydrophilic substances, such as ions and other molecules. This is exactly the barrier that we will have to cross in the next chapter. Okay, now, but biomembranes are not always a plane, as we see here in this electron microscopy figure or in this schematic representation. They can sometimes attain a more sophisticated structures. One example for such structure is the cilium. It's a hair-like structure that protrudes from the surface of the membrane. It's actually a membrane that is very tightly folded. And a couple of hundreds of these cilia, when they move in a single synchrony, they can cause fluid to flow across the surface of the membrane. And this serves for several tasks. Never mind right now what for, but again, it's a good example for the different structures that biomembranes can attain. And another one that's very important for neuroscience is the myelin sheath. So if you ever heard about this structure already, it's actually a biomembrane that covers projections of neurons, neuronal projections. In this figure, you see an axon in a transverse section. Transverse meaning that the axon goes out of the board and into the board, and you have to slice it transversally. So you see it here as a circle. The myelin sheath that covers, that isolates this projection is actually a membrane of another cell, a nearby cell, a glial cell, what is called a supportive cell, so we call it glial cell. In the peripheral nervous system, it's usually a Schwann cell. In the central nervous system, it's usually oligodendroglial cells. And as a whole, these cells approach the projection, and then its membrane starts to wrap itself again and again around the axon to form a very thick and dense layer of lipids. This layer isolates very effectively any leakiness in ions out of and into the axon. And thereby, it facilitates the conduction of nerve impulses over long distances in neuronal projections. It's a great electron microscopy figure. You see here actually the lipid layers one by one after the other. It's very hard to get. Okay, this is a schematic representation that shows the continuity between the different organelles in the cell, and we will in a short while go over each of these organelles. But for now, just notice that, okay, the following facts. So first, the nucleus is covered by a double membrane, not to be confused with the fact that each membrane by itself is composed of two layers. Each membrane is a bilayer, but still, so the nucleus is covered by two such bilayers, by two membranes. They fuse together in certain spots to form a pore. The inner membrane of the nucleus sort of defines the nucleus itself. The outer membrane is in one continuity with another organelle, the endoplasmic reticulum. And so what is called the intermembranal space between the two membranes of the nucleus is actually in one continuity with the endoplasmic reticulum intermembranal space. Okay, do you see it? You can go from here to here, no problem. Mitochondrion is in single, mitochondria is in plural. In Latin, the same for nuclei and nucleus. So I will sometimes use this and that, and that's the difference between the two. Mitochondria have also a double membrane. So they also have an aqueous solution inside them, which we have termed a lumen. Mitochondrion have a lumen, and nucleus have a lumen. Okay, so we also see the Golgi apparatus and lysosomes. We'll go in more depth on these organelles in a short while. But the last thing to note is the two sides of each, is the two sides of the bilayer, and its continuity between different locations in the cell. So, for instance, lysosomes, which are vesicles that bud out or bud into the cell while they engulf some a fraction of the outside substances. So if this vesicle will close to form this vesicle, so now the black line, the inner layer in this structure is actually the exoplasmic side of the plasma membrane. This is where you find the exoplasmic lipids, in opposed to the cytosolic lipids, which are the intracellular lipids. So black here and black here, because you bend this membrane towards internally, and then you get this structure. So, for instance, a protein that will be localized in the internal part, in the intermembranal space of the endoplasmic reticulum, will eventually find itself facing towards the exoplasm. You see this? A protein that will be localized right here, that will be harbored right here in this lipid layer, will find itself eventually facing towards the exoplasm. So this is the way the different organelles are connected between each other. And now we've already mentioned the principal structure of lipids, and now we will go a little bit deeper into this. So lipids can be, they come in three different flavors. They can be phosphoglycerides, sphingolipids, and steroids. And we will now see the structure of each of these subclasses. All of them have the amphipathy feature we've mentioned, the fact that they are also, they have a region of they have a high hydrophobic region and a high hydrophilic region. They differ in their chemical structure and in the characteristics that they give to the membrane. Okay, now the first class, the phosphoglycerides, are all derivatives of a glycerol 3-phosphate, here in red. A glycerol 3-phosphate. So this moiety here will be found in all phosphoglycerides. And now to this basic component, you can play a Lego and link other parts to it as you wish. So you can link two fatty acyl chains with varying lengths and varying degree of saturations via an oxygen, meaning that the two tails are esterified to this glycerol. And you can also esterify to this last phosphate a variable head group, a polar group. Esterified is a link via an oxygen, ester. And again, in the other side, in the phosphate side, you can link polar groups for several identities. And all of these molecules would be termed phosphoglycerides. The basic backbone of this molecule, which is right here in red, this is a glycerol. It's just the way it's built. You have a glycerol and a phosphate, and everything that is built around this basic structure is a phosphoglyceride. Yes?
So we can have four possible head groups.
Even more. These are just 4 examples.
So both sides can be
Exactly, yeah. You can play with this side and with the degree of saturation and the different lengths of the hydrophobic tails.
What does it mean, the degree of saturation?
So, okay, so hydrocarbon tails can be saturated, meaning that they will have no double bonds along them. Remember, it's a recall from previous classes. And in this case, they will be linear. Their shape will be just a straight line. And when you introduce a double bond, then the degree of saturation of the hydrogen across this tail decreases. So you will have some region of unsaturated carbons, meaning that these carbons, for instance, if you introduce a double bond here, will have one less hydrogen, and the same for this carbon. And then this tail will no longer be saturated in hydrogens. It will have 2 less hydrogens than it could have been. And in this case, or this double bond introduces also a rigid, stable kink or bend in this tail. So the structure is no longer will be linear, but will have a bend in it. Okay, so you can play with both these parameters, the length and the degree of saturation, in any fatty acyl chain. The second structure is sphingolipid. So, and sphingolipid has the basic structure, again, here in red. This time, okay, so it's an alcohol, an amine alcohol. This time it has a built-in fatty acyl chain in the principal structure, in opposed to the previous structure. So raise your eyes for 1 minute. So mark this red structure here. The previous one, the phosphoglyceride. So the glycerol does not have any intrinsic stable phosphoacyl chain, and fatty acyl chain, and you can play with these 2 chains as you wish. The principal structure of the sphingosine includes fatty acyl chain. So with this, you can play in this structure, but you can play with the second tail that will be linked to it, and again with the polar groups that will be esterified in this side of the molecule. Okay, so these are the 2 normal-looking lipids, and the third one is a funny-looking lipid, but it's still a lipid. It's a cholesterol. It has a hydrophobic region. So this time the hydrophobic region does not have a hydrocarbon chain. It has aromatic rings in it, but still this region is very hydrophobic, and it has a hydrophilic polar head group, and it exists in membranes, and so this is a lipid. And this is what people are afraid so much from cholesterol. Okay, now these 3 different classes, each one attribute different characteristics to the membrane, and we'll see it in the next slide. Okay, the cell membrane, the outer cell membrane, is also, as you know, called the plasma membrane, So plasma here indicates the fluidity of this structure. It's not a rigid structure, but it's a structure in continuous motion and fluidity. And so most lipids and most proteins are laterally mobile along the surface of the membrane. By laterally, I mean in parallel to the membrane surface. This is what lateral means here. And to get an intuition of how fluid is the membrane, so the viscosity of the bilayer is around 100 times more than that of water, which means that it's around the same viscosity of that of olive oil, if it helps anyone. viscosity is like צפידות, אני חושב בעברית. צמיגות, אולי נכון, צמיגות. Viscosity is the fluidity. I think it's almost the same. Yeah. Sort of swimming in honey instead of water. It's maybe more cozy but less convenient. Okay, so now inside this ocean of lipids that is in constant motion, you find some colocalized lipids and proteins, some sort to speak, so rafts or floating islands, if you wish. And okay, so these are colocalized lipids and colocalized lipid rafts, which also have some proteins interact with them. Yes?
Where are they located?
Where are they? Sorry?
Where are these rafts located?
On the membrane itself. So the membrane itself is composed of components that the location between them is constantly in change. So the lipids are in constant lateral movement in relation to one another, but then I'll tell you, and then I say that some lipids are colocalized, or the interactions between them are stabilized, and together with other proteins, they form such rafts. I mean, so some fractions of the membrane are unmobile or much less mobile than the other fraction, and to these fractions we call rafts or islands. Okay, so they are localized. They are actually part of the membrane. So obviously they are localized in the membrane. Okay, so as we said, lipid composition influences the physical properties of the membranes, and we will now see how exactly this happens. So, for instance, what determines the degree of fluidity in the plasma membrane? What influences how gel-like will it be, or how fluid-like would it be? So there are three components that determine this aspect. The first one is the length of the fatty acyl chains in the lipids. The longer these tails, the more hydrophobic effects will be formed here, the more van der Waals interactions will be found here, and the more stable the interactions between these lipids will be, and the more we will be pushed toward the gel-like consistency of the membrane. And vice versa, the shorter these fatty acyl tails are, the more towards fluid-like consistency would be in the membrane, because less interactions would be able to form. The second component is the degree of saturation. So again, if you have saturated fatty acyl chains, meaning that they have the full number of hydrogens along them, this is the state in which the configuration of the tail is a linear one. It's just a straight line. In this case, again, even the distances between the different atoms will be the shortest, and then the interactions will be the most stabilized ones. And again, we will be pushed towards a gel-like consistency. So now I speak about so again, more saturated fatty acyl chains, meaning a more linear configuration of that tail, meaning more interactions between the tails, meaning more gel-like consistency. And vice versa, if you have an unsaturated fatty acyl tails, these tails have bends in them, right? So these bends just disrupt some of the possible interactions between the tails, and we will be pushed towards fluid-like consistency. Okay? And the third component is actually the presence of cholesterol. So cholesterol pushes us towards a fluid-like consistency. The existence of a high level of cholesterol in the membrane will push you towards fluid-like consistency. And this is why, by the way, Arctic animals have a high degree of cholesterol in their cells because they have to sustain some certain degree of fluidity in their cell membranes in extremely cold temperatures, and this is exactly what the cholesterol allows them to do.
What does the fluidity happen?
Okay, so now fluidity of the membrane, that's a good question, and fluidity in the membrane is essential for several processes that happens along the membrane. It's not an artifact, it's not a accidental feature in the membrane. So for instance, and we will discuss it in more depth in future classes, but for instance, when there is some signal that is transported into the cell, for instance, via a G protein coupled receptor, so some a component of this complex would, after activation, would travel along with the movement of the lipids in this plasma membrane and activate other components which it will encounter, and by that this determine how amplified will the signal be as a function of how fluid will the membrane be. But now too much fluidity obviously disrupts too many interactions between the lipids, and the membrane becomes shaken and not stable enough. So you have to have a certain degree of fluidity to allow for several processes to occur, but on the other hand, not shaking the stability of the membrane as itself. Okay, the lipid composition not only affects the gel-like or fluid-like consistencies of the membrane, but also determines the bilayer thickness. So this is more intuitive, maybe. So this is the side view of the membrane. I've just sliced the membrane from the outside of the cell towards the inside, and we look at it. We are now inside the lipid bilayer and looking towards the side. So this is a side view. So the shorter the fatty acyl tails, the thinner the membrane itself will be, and the longer the fatty acyl tails would be, the thicker the membrane will be in this region. Again, cholesterol also plays a role in the thickness of the membrane. So at the presence of cholesterol, while it interacts with other amino acid fatty acid tails, it sort of can effectively increase their length and cause again a thicker region in the membrane. And the last aspect that is affected by the composition of lipids is the curvature of the membrane. And this is because certain lipids have different general structures in space. So some of them would have a cylinder structure. Others would have a cone structure in space. And now just put several cones inside and several cylinders outside, and you will gradually build yourself a curvature in the membrane. Yes?
And so the thickness of the membrane is not equal to all the lengths, right?
Right.
And what are the functions of a different thickness?
Okay, so now, I would start like this. If you have a high enrichment of specific lipid in specific region in the membrane, it can become thicker or thinner in compared to the other regions. Now, some proteins are stabilized in a membrane. Some intermembranal proteins that are embedded inside the membrane, are stabilized in membranes this thick, and others stabilized in membranes this thick. And so, if you need to stabilize a protein in a specific region, one thing to do would be to stabilize a specific thickness of the membrane in a specific region for this protein to be more stabilized in it. Any questions about this slide? Okay.
The curvature it has no connection to the thickness.
Well, not directly, no. Yeah, for us it's independent. I guess it's not really independent, but yeah, for us it's independent. Okay, lipids are usually distributed unequally between the two sides of the membrane. So there is an unsymmetrical distribution of lipids between the two membranes, meaning that each side of the membrane will have different population of lipids. And why it is so? So there are several observations that are considered to be the reasons for this phenomenon. The first is the different susceptibility to hydrolysis by phospholipases. So phospholipases are enzymes that cleave lipids along them, and by that changes structure. So for instance, phospholipase D will cleave the lipid here and will give rise to a different kind of lipid, which will give rise to a different set of characteristics in this specific region of the membrane, right? So the question of the question is what? Well, what? So I will start again this slide. So let's start with the fact that lipids are distributed unsymmetrically between the two sides of the membranes. And now I try to explain you how this asymmetrical distribution is formed. So there are two observations that have to do with this phenomenon. The first one is the presence or absence of phospholipases, which are enzymes that cleave lipids in several sites along these molecules, giving rise to other molecules. So lipid X will be cleaved by phospholipase D and will be then transformed to a lipid Y. X has a certain characteristics that it attributes to the specific region in the membrane, and when it changes its structure, it has another properties, right? So a curvature can be destroyed or formed, and a membrane can be thicker or thinner just as a function of the set of phospholipases that exist in a specific region inside or outside of the membrane. All right? So this is the first determinant for the different distribution of lipids. Yes?
This is all one lipid?
This is all?
One lipid?
Yeah, this is one lipid.
So if it cleaves a part of the lipid, it's still one lipid?
That's correct, but okay.
Why is the distribution different?
So phospholipase will cleave this lipid and then a close by lipid and another lipid and another one. And when it can gradually, well, you have more than one phospholipase actually. If you get a population of phospholipases in a certain position in the cell, right in front of a membranal region, they can gradually shift the distribution of the lipids from one type to another, right? Because they will change one lipid and then another one and then another one until the whole population will be changed.
So you're talking about one type of lipid that is distributed unequally?
No, so again, so this fact is true as the following phrasing. So as a whole, when you look at both sides of the membrane, you see different sets of lipids. So that's about the population. That's true. That's also true for individual case actually. So for lipid A, it will be found in high concentrations in the inner side than compared to the others, to the outer side. That's true. Okay, so that's true for the population for the individual levels. And now I try to show how this asymmetrical distribution is formed. So first by phospholipases.
So can you repeat why? I mean, they cleave
Lipids,
but
yep.
But why does that affect the distribution of lipids in between the two layers?
Okay, so let's start with okay, so is that sort of what was your question as well? Okay, so
Still didn't get an answer.
okay, so here I'll try again. So if let's start with the initial step in which the two bilayers, okay, the two sides of the membrane had the exact same distribution of lipids. Now let phospholipases X be present in the inner side, in the cytosol, and do not let it be present in the exoplasm. So the inner side of the membrane will be gradually changed to a different kind of lipid, right? Because the phospholipase will cleave it, change its structure, hence change in its characteristics, hence change the curvature of the membrane. You will, for instance, you can be transformed from this lipid to that one. So if initially you had in both sides of the membrane, you had the red cylinders, you can gradually give rise to a much higher concentration of the cones in the inside, in the cytosolic side, and gradually a curvature will be formed. So now the question of lipid asymmetry, I didn't really answer for how is it formed. I actually changed the question of lipid asymmetry by changing it to phospholipid asymmetry between the two sides of the bilayers. Is that okay? Okay. So that's the first determinant, and the second is what is called the vectorial synthesis of lipids. So very briefly, cytosolic lipids are synthesized inside the endoplasmic reticulum mostly, and lipids that are embedded in the exoplasmic layer are synthesized in the Golgi mostly, and the two. So it's enough now for these two organelles to transport vesicles in an opposite orientation for more of a certain kind of lipid to be accumulated in the exoplasmic side, and more of a different kind of lipid to be accumulated in the cytosolic side. Again, the endoplasmic reticulum, this is where most of the cytosolic lipids, meaning the lipids that stands in this side of the membrane, are synthesized. And so the membrane that will be bud out of this organelle and fuse with the membrane, with the plasma membrane, will have lipids in this side, and the Golgi synthesize lipids in different sides, and these two organelles just synthesize different sets of lipids. And this, again, is something that influences directly the distribution of lipids across the two bilayers, the two layers. Is that okay? It felt shaky. Okay. So we will just end this, I think, 2 more slides and then go out to the break. Proteins interact with the membrane in several ways. So they can be integral or transmembranal proteins, meaning that they will have 2 edges sticking in both sides and just embedded in the membrane. So, for instance, ion channels and other proteins are of this type. They can also be harbored to the membrane, but not protrude to the other side. They will be harbored to a lipid, and then they will move together with the ocean of lipids. In this case, they are called lipid anchored proteins, and they can also non-covalently interact with the membrane. For instance, this protein right here, or not interact directly with the membrane at all, in which cases they will be called peripheral proteins, peripheral to the membrane. And now these proteins, in their turn, the intracellular ones interact with the cytoskeleton, and the outer cellular ones interact with the extracellular matrix. It is sort of the equivalent of the cytoskeleton from the exoplasmic side. Okay, and last thing we will mention here is a structure that is very common to transmembranal proteins. Okay, this type of proteins. So first of all, most of them will have an alpha helix. A question: why? Why would transmembranal proteins so often have alpha helix in them? It's a strong structure, but strong in what sense? So first of all, it's just opposing bending very, very effectively. That's correct. And what else? So remember what happened. What are the interactions that stabilize the structure? The interactions are between the polar groups in the backbone of the polypeptide. So most of the hydrophobicity, most of the hydrophilic nature of a polypeptide is already taken or already destroyed solely by the presence of this structure. And now it's enough to have non-polar amino acid groups that face towards the outside for this structure to be highly hydrophobic. And hence it will be highly stabilized inside the lipid membrane. So this is why the alpha helix is found in so many transmembranal proteins. And more specifically, really a huge amount of transmembranal proteins will have a specific structure of 7 membrane-spanning helices, which is the structure depicted here. So these 7 membrane-spanning helices is really a hallmark of transmembranal proteins. So it was worth mentioning it here. It is found in ion channels and G protein coupled receptors and others.
Can you repeat why alpha helix was the
Okay, so let's start, what are the interactions that stabilize this structure? Do you remember?
yeah, the hydrogen bonds between the backbone.
Right. And the most of the hydrophilic nature of a polypeptide is due to the fact that you have so many charges across the backbone. And these charges are already neutral, effectively. For the environment, they already interact with one another, so they sort of neutralize each other. And they will no longer be able to give a hydrophilic nature to the protein. And now the amino acids that face towards the outside here will determine the hydrophobicity of the protein. So, for instance, if this region so you have many non-polar amino acids, right? So just place a couple of them in a specific region, and you will get a highly hydrophobic region faces toward hydrophobic region, which is the inside of the membrane. And so this protein will be, will feel good about itself sitting in the membrane.
Why the amino acids that hydrophobic?
They're not necessarily, but if they are hydrophobic, that's enough. I mean, that's because the hydrophilic nature of the protein is already neutralized, and it's enough to have a few hydrophobic amino acids for this structure to be very hydrophobic. Because when you have a linear structure of amino acids, even if they are hydrophobic, one side of them would be hydrophilic because you have polar groups in the backbone itself, right? But exactly these polar groups are already engaged with each other in this structure. All right? Okay, and we will skip this slide to just give 3 examples of a lipid harbored proteins. It's not not essential. And we will take a 10 minutes break and continue afterwards with the organelles of the eukaryotic cells. So let's take the break.
D is between several perpendicular alpha helices. So then 2 was...
You said
Two
okay,
regular
Felix,
is...
don't worry.
I said
He's like,
D and D.
he's like, don't worry about it. This is not a main question.
Because like the smaller the amino acid, the better the angle will be, right? That's the logic. The smaller the amino acid. So then 3
C.
is not correct.
Not correct. No. Like C is the correct answer because that's the English one. It's not correct.
Oh, right.
That's right.
Forgot about that. Nice. And 4 is
So
the H, which was B and D.
what? 4 C and D. Okay. Yeah.
5 was... I said B or C.
C.
Oh, right.
6?
And 6 was... Oh, right. So what is this happening? Okay, so 6 was H. No,
A and
A and D. So the things that weren't right, that doesn't make sense. Catalytic sites for an enzyme. Oh, no. So wait, A and D. So it is a catalytic site. Oh, where an enzyme binds. And then it's a ligand binding site, but it can't be a membrane. Yeah, that would be a membrane binding site. And binding site for other proteins doesn't make sense. Yeah. Maybe it would be an catalytic. Okay?
7?
I said G. A and B.
Yep, that's right.
Too bad.
Well done.
Do you have that on the room for class 1 by any chance? You got all your notes together there?
It's hot right now.
How are you doing, by the way? Are you still sick?
No. I'm okay. Yeah.
Where were you when the alarm went off?
I was at home. Good order.
You were safe?
I was in my little shelter. I was safe. Where were you?
Also in the... Although it was really weird, I think one of my roommates was a death wish. He was like, he didn't want to go in the shelter.
Really?
Yeah. Like I've never said.
Like our friend Fatima over here.
Really, you too?
She was in the dorms as well apparently and decided not to go to shelter.
Yeah, because I didn't know about the testing.
Oh. But this guy, we were telling him, come in, and he was like, cleaning dishes.
Really? He's like, I don't worry about it.
He's like, I'll come in a second. It was like, yeah.
You have any seconds?
I have 30 seconds just to clean this dish.
That's funny. So, because you're in the dorms from us, right?
Yeah.
And so you guys have a shelter that's right there.
Yeah, no, there's a shelter in every apartment. Like
In every apartment?
there's a mamad, which is like the protected version of the space in each shelter. There's one single shelter which is big, but it's not open.
I don't think I have answers for this.
Oh, it's no problem. אין בעיה.
תגיד, השאלות שאנחנו פותרים, זה יהיה השאלות של המבחן או שזה יהיה דומה?
אני מניח שזה יהיה דומה.
זה לא מאגר שאלות שהוא... כי נראה לי שזה פעם אחת הוא אמר שזה השאלות של המבחן שלו.
Right. That'll be the question for the test. These will be the questions for the test.
I think once he said something like this, or it might be extremely similar,
Right.
or it might be this. So it's like a bank of questions, a lot of questions eventually, until the end of the semester.
Really?
הנה, השאלות שאנחנו פותרים כל הזמן, זה מתוכם לוקחים למבחן או שהמבחן יהיה דומה?
לא, המבחן והשאלות לקוחים מאותו הבנק.
כן.
So it will be.
It's exactly the same.
It's from the same bank, but they will not be the same. It will not be the same question.
Right.
The exact same type, the same style. It's a multiple choice exam. Same level. You know.
They're not the same question.
No.
Not the same.
Not exactly the same. No, not the same at all. Just the same style and the same. But there's always the question of how deep you need to go before doing the test. And so I don't have an exact answer, but the answer should sort of arise from answering along the semester to different questions. So it's just that idea.
I like trying to find different variants. siRNA is really cool because that's the regulation of gene expression, so you can use that to like find which gene inhibitors.
זה בעצם כזה של פרומון?
כן, מי שצריך להיות, או שהוא...
חייב שיהיה פריט
that
לא בהכרח. זה קורס של ICNC, ICNC מחליט.
זה לא קשור למי שאתה עובד אצלו במעבדה.
ב-ICNC לא. שוב, זה קורסים של ICNC, אז גם שגם במכונים אחרים זה ככה. שזה לא הפרופסור מחליט, זה ועדה חיצונית.
Plus, I go to Iowa, the system I want to work with as
SIMA. By the way, did you take public transport to get here? I was going to take the shuttle from Mount Scopus, but they were full. Like, completely full. So I had to take a bus, a train, and another bus. What? Why? Because there's so much traffic, so like I didn't take one straight bus. How would you know there's traffic? Because 7 o'clock traffic or 7:30 traffic. So now here's the worst part.
How would you know how to take this bus because the other bus won't traffic?
No. Taking the train, which has no traffic, it's on the track. Now here's the catcher. There was a mysterious object, suspicious object in the central bus station. So the train got stopped for like 20 minutes. I'm sitting there for 20 minutes, like it's never gonna go. So I got out. I took another 9 bus. I guess it goes it goes through where you live.
So I get here.
Oh right, so I had to take that from the office.
The 9 goes to the South African Gazette?
No, the thing is we couldn't get into the South African Gazette because of the sister shop. So I was stuck at King George.
sorry.
No, I took
Rough morning.
Yeah.
That's why you should get a car.
Or like,
Bike.
you both wrong.
Honestly, I was thinking about getting a bike, but I just don't know how to buy a bike here. I feel like I'll be like fired out. I'll get cheaper stuff.
Why did you decide to stay in Montgomery?
It's just a nice place and live already there. I mean, with the shuttle there, it's like there's no problem. But again, like I never expected the shuttle driver. Usually they let people stand. I never expected him to be like.
The guy said no.
He's like, get out of here. Like
No.
he had no sympathy.
That's bad.
Yeah, no, I mean, people have no situation, but whatever. Also, I can do more whole time. That's nice.
So did you do a home right now?
I'm trying to, but that's also another touchy point. They're like, they're like treating me like a criminal. It's funny.
Why?
It's like, oh, I want to start with fun. I'm like, no, it's too late. But no, like I told them, no, I'm not really taking third grade. I'm just trying to see my degree. They're like, no. And then they're trying to make me take the one for the graduate program. So that one's at 8:30 in the morning. Like, so it's like, it's like, and they send me these emails like, just go get it. Like, what is it? Oh, this lady sent me the first sentence was, you're wasting valuable time.
Wasting valuable time?
Attends, le bed, man, oh no, attends, mais bed, man, get car or something. You're losing time. Yeah, it's even worse than wasting.
What did John say? Time you doing wasting without wasting?
Come on.
She'll sit behind the wall.
You guys, I've only been to one acting class. Out of 4.
The course.
Oh, 3, right. I've been to 1 out of 3. It's really bad. It's about a 6 or the second night.
בואו ניכנס.